Hi everyone, in this video we're going to learn about protein solubility. So whenever we talk about solubility, there are two terms that we have to be familiar with. The first one is solute and then the other one is solvent. So as you can see over here that in a beaker I have put some molecules, right? So the molecules that is getting dissolved in the solvent is known as solute. So for example, sugar, salt, all these things are solute, right? And then when we mix that in a water, in the water, the water is actually the solvent. So solute is actually the molecule that is going to get dissolved, and solvent is actually the molecule that will allow the solute to get dissolved with itself. And then together, we call it as a solution. So I've just written a short form. So as you can see over here that whenever solute and solvent will mix together, they'll be termed as solution, right? Now let us try to understand the protein solubility. So protein solubility is it's not that of a complex topic. We'll try to understand it very quickly. So as you can see over here that whenever a solute will dissolve in a solvent, that means this solute that I'm talking about is actually a protein, right? So this protein is actually getting dissolved in a solvent. The solvent could be anything. Now for example, the protein is getting dissolved in a solvent, and the protein that is dissolved is spread in a very like in a very equivalent manner. That means the solute has been spread out in a very equivalent manner. Then we can say that this is a properly solubilized protein, right? But in this case, as you can see over here, that the proteins, rather than to form bonds with the water and be dispersed equivalently, it seems like the protein has kind of like formed bond within themselves. That means they have formed an aggregation of their own molecules. So can I say over here that the solubility over here is high, right? That means the solute is dispersed, right? And the reason why the solute is dispersed is because the solute is actually forming bonds with the the water molecules, right? The protein is forming bonds with the water molecules, therefore it is equivalently dispersed properly. But over here, as we can see that the protein is not forming bonds with the water molecules, right? And therefore it has form and aggregated form. So this particular form is actually known as the precipitation. So whenever you must have learned about certain molecules, certain, like in chemistry you might have learned about the term precipitation or any term in scientific term. So this is actually the meaning of precipitation, right? So whenever any compound is not able to form proper bonds with water, and then it tries to clump itself and then tries to interact with one another, and therefore it forms precipitation, or we can say it forms clump. Now, what do you think the solubility in this case would be? Because over here the solubility was quite high, right? Because the solute and the water molecules were forming bonds with one another very properly. But then over here, as you can see that the solubility will decrease. That means, can I say that it will decrease? Yes, the solubility has decreased. Decrease, okay. And can I say that the solute is dispersed equivalently? No, right? It has aggravated. So I can say that it is kind of like forming clump. So I'll just write it out here. Forms clump. Okay, so now let us go deeper. So now what exactly is precipitation? So as I told you that precipitation is when the solubility is really low. That means the solute and the solvent is not able to form proper bonds with one another, right? Now I have taken the example of solvent, in this case water. The solvent could be anything, right? So just a common example, that is water. And then there is formation of clump. So they will form, they will form interactions with one another, and therefore they'll form a clump. And then whenever we talk about relation of precipitation with solubility, there is an inverse relationship. That means, as you can see over here, I have written that solubility is, inversely proportion to precipitation. As you can see over here, when solubility is increasing, precipitation is decreasing, right? And then when solubility is decreasing, precipitation is increasing. That means they are inversely proportional to one another. Now, when we talk about solubility, there is a very there are many factors which influence the protein solubility. Now there are certain intrinsic factors and extrinsic factors. Now intrinsic factors are actually the factors which are which does not include the other environmental factors. For example, it is just taking consideration of the protein like itself. So intrinsic factor means, for example, amino acid composition and sequences. That means, the protein solubility is influenced by what kind of amino acid is present in this particular protein and what kind of sequence is also there, right? And the other one is molecular weight. That means if a protein is a larger molecule, then will it be soluble in the solvent, or if it is a smaller molecule, then will it be more soluble? So it depends upon the size as well. And then the third one is conformation. So conformation means, like, either it is in the globular form, either it is in the unfolded form, so unfolded form means like the primary structure, right? The primary structure. And then globular form means the tertiary and the quaternary structure together, right? As we have already studied this in our previous video in hemoglobin, what exactly is a globular structure, right? So let me just take and make it bigger. Okay. And then there are extrinsic factors as well. We're going to talk about it later. First, let us just look at the intrinsic factor. When we talk about amino acid, there is a classification upon what kind of R group does an amino acid possess. So if the amino acid has a polar group in its R group, then it's going to be a polar molecule, right? And if it does not have any polar groups, then it's going to be non-polar. So in simple language, I can say that non-polar groups means like hydrophobic, and then polar groups means hydrophilic. That means this is going to carry a charge, and this is not going to carry any charge, right? So if a protein has polar group present in its R group, and then as a whole, if the like amino acid is a polar amino acid, then it's gonna be more soluble in the solvent, right? And then if the amino acid does not have any polar group, that means it is a hydrophobic amino acid. That means the the amino acid will not be able to properly solubilize with the solvent. So if I just have to write over here, then I can say that if I say polar, that means in polar groups, the solubility will increase, right? And then if I talk about non-polar, then the solubility is going to get decreased, right? And I've made a list of the amino acids, about the polar and the non-polar group as well, right? So as you can see over here that these are the non-polar groups and these are the polar amino acid. Glycine, alanine, valine, leucine, isoleucine, proline, tryptophan, phenylalanine, and methionine, right? And then over here we have D as aspartate, we have glutamate, and then we have histidine, then we have arginine, we have lysine. So if a question comes like, this particular if D and E amino acid is being, it's being like put into a solution, and then sorry, in a solvent, and then a solution is being made, then what do you think the protein solubility will be? So the question can come like this. So you can say that the protein, since the DNE the DNE amino acid is a polar molecule or polar group, then the solubility of the protein in the solvent will increase. Okay, so let us go to another one. So when we talk about another one, that is the molecular weight, right? So molecular weight means like if the protein is a larger protein or a smaller protein, right? Sometimes for example, if a protein consists of a very like if a hemoglobin is compared with myoglobin, then definitely we can say that hemoglobin is more heavy than myoglobin, right? Because it is a larger structure. Just a simple example. But a larger protein is very unusual. It is very unusual for it to solubilize properly. Now I can write it down that when a protein is very large, right, then what will happen is the solubility will decrease. And then when the protein is, let me make it small, is really small, then the solubility will increase, right? Now why do you think this would be the case? It's because sometimes what happens is a larger protein can have a larger surface area, right? If you compare this surface area and then this surface area, this is a larger surface area, and the larger surface area could have more hydrophobic amino acids present on its surface. That means I can say that the amino, let me take another one. So I'll just take purple. So I can see that sometimes what happens is larger the area, surface area, more hydrophobic amino acids present on the surface area. That means the surface area is exposed very much, therefore there is a possibility that hydrophobic amino acids could be exposed, and therefore when anything hydrophobic is being exposed, then obviously solubility is going to decrease, right? But let us take this example. So when the protein is a smaller protein, so what will happen is the hydrophobic area will be less exposed, right? So I can say that this is a less exposed hydrophobic amino acid. Now whenever we talk about protein, there is a possibility that sometimes what happens is the hydrophobic area is inside the protein. That means whenever protein is folding, there is a case that the hydrophobic will get inside of the protein, and then the polar part or the hydrophilic part will get outside of this surface. That is the case, right? But sometimes what happens is when larger surface area is exposed in hydrophobic amino acids, it's not mandatory that all the amino acid which is present on the surface that is going to be polar in nature. There is a possibility that hydrophobic and hydrophilic both are present on the surface, right? But hydrophilic is more in number. That's why whenever we talk about a protein surface area and then the core area, this is the core area, right? So the core area is usually the hydrophobic part, and then the surface area is usually the hydrophilic part, although they both contain hydrophilic and hydrophobic, but the more in number, that's why we call it as hydrophilic, this one, and then hydrophobic, this one. So I hope you understood about the molecular weight as well. Now let us try to see the conformation of protein. So the protein can either be a globular protein, that means a folded protein, or an unfolded protein, right? So a globular protein, have, I'll just write it down over here. Globular protein has a chance of less solubility. And then when we talk about the unfolded protein, solubility is more. Now, why do you think I am talking about like this? Solubility is more. Because when a protein is being folded in a globular structure, the hydrophobic part, most of the hydrophobic part gets inside this core part, and then maximum of the hydrophilic parts goes outside, But in terms of unfolded proteins, what happens is, in unfolded proteins, the hydrophobic and the hydrophilic part is both exposed. It is not folded, no, it is like in a linear structure. So like the hydrophilic part, all the hydrophilic part is being exposed in the long proteinaceous chain. So rather than the globular, the unfolded structure of protein is more soluble in the solvent. As you can see over here, that the surface is polar and then the core part is non-polar, right? Now let us try to see some of the extrinsic factors. So extrinsic factors means like the environmental factors, like the pH, temperature. So we're going to learn about two. So if you do not know what exactly is an PI, I request you to go back to my previous video and learn what exactly is PI. But let me tell you in a very short form that PI is actually the pH, where it is an isoelectric point. So whenever a protein reaches a certain pH, it carries a total net charge of 0, and that is actually the isoelectric point. That means the point where it loses all its charge and becomes totally 0, like net charge 0. So that is actually PI. So whenever a pH is equals to PI, then what do you think? The solubility will increase or decrease? Just in terms of PI, it will decrease. Why do you think it will decrease? Because isoelectric point has a charge of 0, and I told you that whenever a polar, if just to take an example, if I take polar and then non-polar, then which is going to get solubilized more? It's going to be polar, right? Because non-polar does not carry any charge, right? So polar is going to have more solubility in the solvent. So since the pH is equals to PI, means that the protein does not carry any charge, that means the solubility will decrease, and then there is a high chance that precipitation will take place. Okay, now whenever the pH is above PI, right, the charge is going to be, the amino acid is going to carry a negative charge. And then if the isoelectric point, if the pH is below the isoelectric point, then it is going to carry a positive charge. Now I just will tell you in very shortcut. Above PI means a basic solution, and then below PI means an acidic solution. When it is going in a basic solution, it is going to donate its hydrogen, right? So it is going to lose its positive charge and become negative. Whereas whenever the the pH is very less, that means it is having hydrogen ions more, that means it is not going to lose any hydrogen ion, rather it's going to gain hydrogen ion, and then that will be contributing to its positive charge, right? So if you still don't understand, you can go back to my video and like study what exactly is pi, but I just wanted to tell you what exactly does it mean over here. So what do you think, at which point does the precipitation takes place? So definitely the precipitation will take place at which the pH is equals to pi. That means the precipitation will take place where the isoelectric point will be present, right? So I hope you understand. Above pi, that is like high pH, and then below pi means like low pH. So I'm just gonna write it down, so if you don't get it, this is high pH, that means this is a basic pH, okay, and then this is low pH, this is a acidic pH. Okay, so now let us try to understand this graph. Very simple graph. As you can see over here, that the solubility is over here and then the pH is over here, right? So whenever when we talk about the pi point, as you can see over here, that the solubility is actually decreasing. At first it is normal, and then it's decreasing, and then when it reaches a net charge of 0, that is the pi, that is the isoelectric point, it's going to get like very low. The solubility is going to get very low, right? And then again, if the pH is high, then it's going to get like solubility is going to get increased. Now when the pH is low, that means it's going to have a positive charge, right? And then when the pH is high, it's going to have a negative charge. So as I told you, that positive and negative is actually that means the compound is actually showing a polar nature, and then if it's showing a polar nature, then it's going to be high in solubility. So when we talk about the pi below and then above, then it's going to be, then the solubility is going to be high only. So it's going to be high at this side also, this is going to be high in this side also, only the pi will not have any effect. So beta lactoglobulin is actually an example that is taken over here to show how exactly the isoelectric point affects the solubility of a protein. And as you can see over here, the beta lactoglobulin has a pH of 5.3, which will show its isoelectric point, right? And then at the pH of 5.3, it loses all its charge and becomes totally least soluble in the solvent. Okay, so this is one thing. Now let us go to another one, that is heating. So now when we talk about heating, we must whenever we want to mix something, for example, if you have added some masala, or if you have added some sugar in your in your solvent, in your water, then when you like heat up the gas, what happens is usually the sugar molecules will get dissolved very easily, right? It's going to get dissolved very easily. So that is also going to be similar in terms of protein, but protein has a little bit of different story. So let us try to understand over here. Okay, so now as you can see that when the temperature, this is the temperature x-axis and solubility is in the y-axis, right? Now whenever the temperature is being increased, the protein solubility is increasing, and then it is reaching a certain point. That means it is reaching around 40 to 50, right? And then suddenly what happens is, instead of going this way, the protein has started to show decrease in the solubility. Now it has been decreasing in a very steep manner. That means it's going this way, right? It is not going like this, and then like this. It's not going like this. It is rather going it is increasing in a very steep manner, and then decreasing in a very steep manner as well, right? So until and unless a protein reaches around 40 to 50 degree centigrade, the protein is going to increase its solubility. That means it is going to like nicely dissolve in the solvent, and all of a sudden when it reaches a certain point beyond 50, then what happens is it starts to denature. That means, the like for example, this is the structure of protein, and then it has folded itself. Right now the folding will, after this structure will try to become like this, right? And then it will try to be a linear structure. Then after linear structure, what will happen is it will try to break the peptide bond itself, then it will become like separate amino acid. So it's going to denature. That means whenever, a protein is exposed to a very high concentration of temperature, then sorry, high like degree of temperature, then the denaturation process will take place. So I'll just write it down over here. Denaturation of proteins. And I told you that, the protein will first, it will try to break the other bonds like the hydrogen bonds, the sulfide bonds, and then finally it will try to break the amide bonds or the peptide bonds as well. So that is the reason why, the solubility will decrease like all of a sudden. Now what exactly is the kinetics behind, the solubility? That means the increase in the solubility in terms of heating when we talk about the protein. So I have written over here. So as you know that whenever heat is applied to any kind of compound, the molecules will start to move in a very like fast manner, right? So simple, if you are touching something very hot, if you're cooking something and you touched very hot, then what will you do? You'll just remove your hand from that particular like utensil or like warm thing, right? And then you'll try to move yourself very fast away from that particular thing. Same with the molecules as well. So the molecules will try to, it has a very high movement at the moment when it is exposed to high temperature. So as this pink one is actually the proteins, and then the blue one is the water. So I'm just giving you an example of water. The solvent could be anything other than water, but just a general example. So this molecule, this pink one, can move in this direction or in this direction, right? And when it moves in this direction, it will push this pink one, and then it will, collide with this water molecule, and it will form a bond, right? Similar with this one also. This can push this one, then this can form bond with this one. Similarly, this can come and this can push this, and this can form bond with this, right? And then this can push this and this can form bond with this one or this one. So it is all about kinetics. So the solubility, kinetics behind solubility is actually the high amount of movement between different molecules, especially the protein molecules, that is, like colliding with one another. So they are colliding with one another, and then finally they are forming bonds. That means more we apply the heat, more they will collide with one another, and then more they collide with one another, they'll form bonds with one another. So this is about protein solubility. So we learned about kinetics, and then we learned about, what happens when a protein is heated, and after a certain point, what happens, it completely like denatures, and then the solubility decreases like all of a sudden, right? Then we studied about the isoelectric point. So isoelectric point is actually the particular pH at which a particular molecule will lose its charge, and then the precipitation will take place, right? And this particular precipitation is known as isoelectric point precipitation as well. So if you want it, you can just note it down. As you can see that below pi and above pi solubility is high, at pi it's very low. So it's going to give a V-shaped graph, which is actually a parabola. And as we know that conformation of protein, then we talk about the intrinsic factor, that is the conformation of protein, the globular and the unfolded one, right? The exposure of the amino acid plays a very important role in this one. And then we have the molecular weight. Larger protein does not get dissolved easily, smaller protein gets dissolved very easily, therefore solubility is high. And then we have amino acid composition, non-polar groups. You can just remember this code. And then over here polar groups. So 1 2 3 4 5 6 7 8 9, right? 1 2 3 4 5. And then we have what exactly is precipitation, how does precipitation forms, and what exactly is the relation of precipitation with solubility. Then you know then we know that whenever precipitation is formed, rather than to make a bond with the solvent, the solute will start to bind with one another, aggregate with one another. That will lead to the clumping or precipitation. So this is all about protein solubility. If you like this video, please like, share and subscribe. Thank you so much for watching.